Welcome back. Today we're going to have a peek at the immune system, and I say peek because you could easily have a course with 32 lectures just on the immune system. And so we're going to get an overview, some definitions, and see how the immune system basically works. The immune system is a body response against foreign invaders. This is a system that wants to recognize self from non-self, and wants to then either inactivate, destroy, or get rid of that foreign substance. It's a very, very wide-ranging system. It's also very, very specific in most of its attributes, so that it has quite a bit of accuracy in determining exactly what is non-self. And it has a peculiar characteristic in that the immune system is extremely tolerant to any substance it encounters during embryologic development. So if you expose the developing embryo or fetus while it's in the uterus to any foreign body you want to, it will later recognize that foreign body as self and not know that it's a foreign body at all. The immune system also has the ability to maintain a memory for these foreign substances once it recognizes them. It differs from some of our non-specific defenses because we have other defenses that are not as discriminating and are much more just basically physical barriers. For example, the skin. It's a waterproof barrier that can, in large part, prevent the entry of viruses, bacteria, parasites in some cases, and chemicals, things that are clearly not wanted inside the body. Our respiratory tract, the lining of our intestine, both of which are exposed to the outside, have a certain capacity to physically block the entry of foreign substances. And for example, the inside of our nose is lined with mucus to catch dust and pollen particles and keep them from getting any further in. So that's the part of the system which is very non-specific. We're going to ignore that almost entirely. We have some basic definitions. We need to look at something called immunocompetence. And that's the ability to function as mature cells in the immune system and carry out those tasks. If we have the right cells in our system, and that system can then do what it has to do to protect the body, that is an immunocompetent patient. We have patients who are either born incompetent or become incompetent because certain diseases have depressed their immune system. The main cells of the immune system are what we call the white cells or lymphocytes. And these cells destroy and detect foreign invaders in various ways, which I'll tell you about. We have a couple of other words, antigens and antibodies, which I think you've almost certainly heard about. Let me show you what a schematic representation of an antigen would be in our first slide. It's defined as the molecule or the foreign substance that provokes an immune response from the body. We're going to have a little bit of a circular definition here with each one depending on the other, but I think you'll see it works. This is an artist's rendering of what an antigen might look like. It could be a grain of pollen. It could be a simple molecule. Basically, on the surface of these molecules are geometric arrangements, and these geometric arrangements provoke and tell the body that this is a foreign substance. It recognizes certain attributes in this antigen and sees it as foreign. Now, the antibodies are the molecules in part of our immune system that react with the specific antigen to kill or neutralize them. This one's a little more complex. This is a molecular model showing one of the specific antibodies in our system. And it shows what I want you to see here as a very specific geometric arrangement. I keep talking about this a lot, and our proteins having this geometry that's vital to their function. If you take this whole molecule or series of atoms and molecules and break it down, it does have functional and non-functional segments. We have this antigen binding site. This is the literal place in the molecule that has a hinge region. It can change its geometry, and it binds to the antigen. Here's another set which doesn't tend to change its geometry. These are also made up of things called light and heavy chains. They're extremely complex in their structure. Now, we have differences in our system in what we call two basic subdivisions that are called the humoral and cell-mediated systems of antibodies. If we look at the next slide, if you look at this picture and divide it down the middle, when I'm talking about everything here to the left, I'm talking about cell-mediated immunity, and everything on this side is called humoral-mediated immunity. And by humoral, we're talking about something that floats in a fluid. These are the components of a system, and the system works together. Now, we have, first of all, to go back to the bone and the bone marrow. In the bone marrow, we have the development of two kinds of cells. When I was in medical school, we only knew about lymphocytes and a few other cells. We had no idea that there was a division in them. I mention that because I want you to know that you cannot look under the microscope and tell one kind of lymphocyte that I'm going to show you from another. They're physically the same. Even an electron microscopy won't show you this. You have to use antibody studies or chemical studies to detect this. So it's a relatively recent occurrence that we know that there are more than one kind of lymphocyte. If you look at this long bone, in the marrow of the long bone, we make cells that mature either in the bone itself or in the thymus gland, which is in just below the neck under the breastbone. The thymus, which is shown here, is actually a small gland, very fatty. It involutes after the first few years of life, and it's actually difficult to find in the adult, but it's there. It resides, as I said, under the sternum, and the certain class of cells come out here and mature in the thymus. So we've called them T cells. That's where they get their name. And before they go into the circulation or into the tissues, they live in the thymus where they mature and become the adult, fully capable, immunocompetent cells. There is another class that matures in the bone marrow, and it's called the B cell for that reason, and it migrates out into the immune system when it's called for on the humoral side. Now, if you look at the cellular side of this, basically, the T cells, which have come from this stem cell in the marrow and mature in the thymus, have antigen receptors that develop on the surface of their cell. And the T cell is activated by T cell receptor recognition of the antigen, that little spiky molecule I showed you. The thing that is interesting and important here is that all these receptors are unique and they number in the millions. They're all specific for different kinds of antigens, so we have millions and millions of different kinds that require recognition. How does it do this? It's very similar to the gene shuffling. We have these recognition proteins that are shuffled and recombined in millions and millions of forms, and we only keep a few of them around. We don't keep lots of these cells on hand, just a few of each kind of clone. So what happens is we get this unique recognition molecule that's sitting around waiting to see one of the possible kinds of other proteins that are going to come into the body and react with it. Now, if you look at the slide, we have these mature cells. They're both called T cells, and they basically break down into two kinds. What we call CD8 positive cells and CD4 positive cells. Look at those as just names. They happen to be the names of the antibody characteristics, antigenic characteristics of these cells, but let's just call them the CD8 and CD4 cells. And they will mediate responses that are directed against intracellular pathogens. things that happen in other cells or by other cells. So we have pathogens such as viruses, which are intracellular parasites. They have to live and work inside cells. Some cancer cells, which themselves are seen as foreign body, and tissue transplants. When we bring cellular material from another body and put it in our body, it can be recognized as foreign unless you're an identical twin, and only if you're an identical twin. This is the cell-mediated response. Now, these two cells get activated. They are activated by a second co-stimulator, a second molecule that's needed to react and prep this cell to get ready to do its job. These chemicals are called interleukins. And interleukin comes from the word to speak between white cells. This is a conversation between white cells which upregulates the activity. They're also called cytokines because there's a cellular conversation that creates a movement or an activity that's energy dependent, and something active is going to happen. The second signal is made because we need a fail-safe backup. When the immune system overreacts, as in an allergy, which is a mild form of overreaction, or something called anaphylactic shock, in which the patient can be killed by an overreaction, somebody severely allergic to penicillin, who only gets a small dose, can actually have the whole body react so violently that that patient can close down their bronchioles, go into shock, and die. That's an overreaction of the immune system. We don't want this to happen. So we have a safety here, and that's the necessity of the interleukin to be called into play. It's as if you had a car engine. You have to, when you get in your car, put in the key and turn it, but your car doesn't go until you shift into gear. Now that's there for a reason. You don't want an engine to take off as soon as you turn the key. It's a safety mechanism, and the same thing happens here. We don't want anything to happen until the second signal is called into play. So after recognition and co-stimulation, the T cell is now sensitized and begins to replicate. It recognizes the invader, and it's going to replicate or reproduce as quickly as possible to make millions and millions of similar cells from that one T cell, or T cell clone. And that takes time. Look back again now at our picture. Here is the mature T cell migrating into position to do its job, and it's going to be activated, and it's going to need activation for another kind of T cell, the CD4 cell, which is the T helper cell. This cell is the one that's going to activate the cell and turn it into a killer cell. It activates the CD8 cell, which up till now was not yet a killer cell. It's also going to send some help over to the other side, but we won't talk about that yet. And this cell is going to leave the lymphatic tissue, the lymph nodes, the spleen, some places in the liver, those Peyer's patches in the intestine, wherever it's residing now after it's left the thymus and gone into this lymphoid tissue, as we call it, and it's going to go out into the body and find the attacking invader, and it's going to kill it. These are all protein actors in this little play that are going to do the work and move the cells and make them recognize each other. So the helper cells produce interleukin-2, and that's the activator, the co-stimulator, that's the second step in this. What happens here is that we only have a few cells that are working. It takes time to reproduce them and make them work. That's why the first time you are challenged, whether it's humoral or cellular, you're challenged with an outside invader, for example, measles. Your body has to respond, make the appropriate response. Meanwhile, you're sick. That virus has reproduced while your immune system is tooling up. But at the end of a week or 10 days, you find you're getting better. The reason you're getting better is because now you've built either the antibodies or the cellular response, depending on which system we're working in, and you're finally able to overcome, neutralize, destroy, and get rid of the invader. When that's done, these cells are going to go away. They're going to dry up, shrivel, and go away, except for a few. that have branched off and are called memory cells. Those guys are going to hang around. And those memory cells are going to be there for either decades or your whole life. Some have shorter life than others, but they're going to be there for a very long time. The next time you get challenged with the measles, taking care of your child, the reaction is going to be so fast because the cells are already present, tuned up, and ready to go, that you will actually kill the measles virus or whatever other one we're talking about before you even get a symptom. So you're sitting there caring for this other person who may have the disease. You'll get that disease, you'll get the virus in your body, but nothing's going to happen. Your body is going to go out and kill it in what is known as an anamnestic response or an accelerated response. And if you look at the kinds of responses we see normally, here is the first exposure and the response of two kinds of antibodies. Patient gets a response and then it goes away. This is experimental and they're just measuring the levels of the antibodies in the blood. And then 28 days later, after you have your memory cells established, if you give that patient a second exposure, look at the rapidity and the height of the response. The speed and the magnitude of the response are markedly increased to help you get over this problem. Now, the cytotoxic T cells, or killer cells, they recognize these foreign invaders, and they go out and they're going to do something to the virus themselves, or the cell in which the virus is living, or the transplanted cell, which it knows doesn't belong in your body. And they're going to go out and kill them. And they do it by what's called the kiss of death. They literally have to go out into the tissues and touch that cell or that foreign invader. What happens is we have here an activated cytotoxic T cell. This is the killer cell. And it recognizes, here's the antibody on a virus-infected cell. It has an attachment and recognition set up here. Here's the antigen MHC1 complex. I'm going to tell you about that in a minute, which is being shown to the T cell, literally presented to it, held up on the surface so it can be seen. It attaches here, and this sets up a process that tells the T cell that we're ready to go. It's literally touched the other cell, the foreign invader, and it will release some interferon, which is another one of the helpers in this system, a molecule that's very complex and activates the big eaters, the macrophages, to come to the area. But more important, it has a killing mechanism that involves two kinds of substances, one called lymphotoxin and one called perforin. These toxins eat their way into the virus-infected cell. The virus has entered that cell as an obligatory parasite. The virus only has DNA inside it. It's a protein coat with DNA and no energy machinery. It needs to get into the cell to use and parasitize the cell's energy mechanism, its chemicals, its DNA, and replicate, and then maybe make 1,000 viruses which burst the cell and then go on to infect 1,000 more cells. That's how viruses work. They're intracellular parasites as opposed to a bacteria, which lives outside cells mostly. Now, this virus isn't going to get that chance because before it's set into motion, its molecular mechanisms that reproduce, you can see here, they're suggesting the nucleus is intact, so the DNA hasn't been injured. The perforins will come in and open this cell, and cytolysis will occur. The cell will lyse. It'll break apart, it'll fall apart, it'll spill out its nucleus and its cytoplasm into the environment and be destroyed. The lymphotoxin kills the systems of the invader, and this is called a lethal hit, or as we said, the kiss of death. Now, they can detach after this contact right here and move on to kill again. Many cells in the body, once they do their job, they're gone. This one doesn't, it is very economical, and we can use this lymphocyte to go around to one after another cells until its life is up and kill again. They also, as I said, secrete gamma interferon, another very important molecule which is going to do the cleanup process. The gamma interferon is going to have the macrophages out there to perform a cleanup process and get rid of some of this foreign detritus and get rid of it out of the body completely. There's another set of cells that are in the system called the suppressor T cells. Suppressor T cells cause downregulation of the whole immune response. We don't want this response to go on too wildly or too long. We just want it to get the job done, and somehow the suppressor T cell knows when to tell the other T cells to ratchet down, to downregulate it by messages which we don't understand. Very little is known about these. One of the important areas is in cancer physiology. Cancer cells, when they mutate and become cancer, happen to have the ability to activate the suppressor T cells. And what they're doing is trying to sneak through the immune surveillance. The cancer cells, which may be seen in some cancers, actually the minority of cancers, but those ones that are seen by the immune system and might be attacked by the immune system, faces an immune system that has now been downregulated because it has signaled the suppressor T cells to downregulate the whole immune system and let them sneak through. Now there are the memory T cells, which I've already told you about. They are a subset of that original clone. They have come out here after activation and hung around as the memory so that they'll be ready for the next time. So once you've seen this foreign invader, you're going to see it very quickly the next time. That's cell-mediated immunity, mediated by lymphocytes, interferon, and interleukins, and attacks cell against cell. Let's look now at the humoral immunity that occurs on the other side. We take that term from the really old-fashioned centuries-old terms that the medical profession used. Vague fluids and vapors were called humors. So these are the vapors of the fluids of the body, including blood. The B cells, which have matured in the bone marrow, are now out in the circulation here, are directed against extracellular pathogens, mostly bacteria, and some viruses that are on their way to the cells, but you can think of them as a bacterial force, antibacterial force. What happens here is here are these guys with their antigen receptors of different shape. They're out there, small numbers but millions of combinations, looking around for invaders, and they're kind of hanging out in the lymph nodes, in the spleen, the lymphatic tissue, waiting until they can get out in the tissues, unlike the lymphocytes, which are wandering around looking for trouble. They're activated by the recognition of a specific antigen, and they too are activated and help boost that secondary response by the interleukin 2 specifically. And they then change into a whole new kind of cell called a plasma cell. Now, this is a cell we can recognize on a blood smear. It looks different. It looks a little bit like this. It's rather smooth, stains a purplish color, and we can see plasma cells in the circulation. They secrete antibodies. They secrete that very complex molecular structure, which I showed you here, that is now going to circulate out in the fluid and going to attack the foreign antigen. They supply millions of molecules per cell per second. This is major event in terms of numbers. They are co-stimulated by interleukin 2 and interleukin 4 and 5 from the T cell helpers, and they're out there and they're going to try to get out and physically attach to these foreign invaders and destroy them by their physical attachment. The duration of this plasma cell response is usually up to about a week for any one cell, and then that cell dies. So these have to be made over and over again. Then we have the memory B cells, which are just like the memory T cells, and they give us the anamnestic or accelerated response the next time we see an invader. And each clone of the plasma cell is from a specific clone of B cells is going to secrete a specific antibody for a single antigen. It's probably the most specific system in the body for zeroing in on one kind of invader. Now, if you look at certain cells, there are lots and lots of differences between the kind of invasion they may undergo. For example, viruses need to replicate inside a cell. The malaria parasite gets only inside red cells. And then the extracellular parasites work in the tissues. You'll see streptococcus infections. The origin will get red and you'll see streaks going up our lymphatics. Or staphylococcus will spread through your tissues and create a big redness there. But they're working in between the cells most of the time. They destroy cells, they kill cells, and they create pus, which is white cells and dead cells. But they tend to be extracellular. The way these antibodies act, now that they've latched on, there is no kiss of death. They don't have that ability. They don't have the perforins and the lymphotoxins. So what do? They actually agglutinate and precipitate. For example, what they do is they will grab on, which is agglutination, and cause everything to come together and precipitate, meaning drop out of solution this now heavier new particle. That's one way they work. And those are ready to be eaten up. They can immobilize a ciliated organism. You have bacteria that have tails like sperm. They can make that tail stop lashing, and then that organism loses the ability to move and is an easy target. They can neutralize toxins from the bacteria. Many of our bacteria kill you by releasing a toxin. The one you probably are most familiar with is tetanus toxin. When you get an infection from a bug called tetanus, which is very common, in fact, ubiquitous on farms, in the field, they hide and live in farm animal feces, so the soil is full of tetanus organisms. It's an anaerobic organism. It requires no oxygen to be present in its atmosphere when it's at work in your tissues. So that's why it happens when you step on that dirty nail. You get a puncture wound that goes deep inside your tissues, dropping off the bacteria in an area that may have poor oxygenation, and then the organism can grow and release its toxin. We always talk about the rusty nail. There's nothing special about rust. It's just it's usually been in a dirty place. Most commonly around farms, country, dirt areas, agricultural dirt areas. Now, we almost never see tetanus. I don't know any doctors who've ever seen a case of tetanus. Mine have only been in textbooks because we've wiped this out with immunization. We have a shot called tetanus toxoid, which everybody gets when they're little and get boosters later on through life, that release an antibody in memory cells that can get rid of the toxin, can neutralize it, and can happen long before you get any symptoms. You heard me talk once about tetanic contractions of muscles. Tetany is the repeated rapid contraction of muscle. Tetanus is named that because it's a central nervous system stimulant. And the patient gets rapid, uncontrollable tetanic contractions of muscles, especially in the face. And the sign of this is called rhesus sardonicus, the sardonic smile, because the teeth are clenched, the jaws are locked together, they can't even open their jaw, and they have a smile. So they look just like that. That's the rhesus sardonicus, that's the diagnosis of tetanus. And these patients die in large numbers once they get the disease. You need to treat this and prevent it rather than give it, or open the wound and clean it out before the patient can develop this disease. The bacteria has to grow in the tissues. So careful what's called debridement, cleaning out of wounds, even cutting away tissue, to expose the tissue to the air and let it heal from within is the way we stop this. That's one of the many ways that these antibodies work. They can also stop viral entry into a cell. They can prevent the virus from protecting it using their antibodies. They can increase phagocytosis, because they can coat the bacteria in something that attracts the macrophages and phagocytes to eat those bacteria. They are very, very specific, and in fact, we use them, for example, in science outside the body called monoclonal antibodies. If we take a particular antigen, for example, one that is carried on the surface of a particular cancer cell, we could isolate it. We could inject animals with that antigen and have that animal's immune system raise antibodies against that specific antigen. That's a monoclonal antibody. It comes from against one antigen from one kind of cell. We could then take that monoclonal antibody and inject it into a patient and tag it. We could tag it with radiation, and it would go to a cancer cell, because that's the only place it would stop. It would find the antigen, and we could light that cell up, or group of cells, with these monoclonal antibodies and see them with a Geiger counter. We could literally, and do all the time, tag them with a color dye, inject it into tissues, and look for the color under the microscope. Better yet, we could load them up with more radiation, attach it to the monoclonal antibody, just the way we did with I-131 for the thyroid, and send the antibody to the cancer cell so that that little bit of radiation, which is a lot in a local place but not very spread out, could then maybe kill the cancer cell directly. And today we're even loading these monoclonal antibodies up with chemotherapy molecules and trying to send them directly to the cancer cell that we want to kill, rather than having them go through the whole body and get the patient sicker than they need be. This is a huge area. We use monoclonal antibodies in detection and in treatment of all kinds of laboratory and clinical problems, including finding out which are the lymphocytes and what kind they are. We then use monoclonal antibodies. Let me tell you about one more kind of example of human activity that gets us in trouble and where we use our antibody system, and that's rabies. Rabies is a virus that attacks the central nervous system. It attacks and lives in the brain, causing death by the damage to the brain. It is often carried by what we call a sylvatic host, meaning animals that live in the forest. That's why we can't get rid of it. We can quarantine all our dogs, immunize all our dogs, but as long as bats and foxes and rabbits have them, we can never get rid of the host. This is why if you go to England, New Zealand, Australia, places where they are islands and can control this and don't have a sylvatic host, you can't get your dog in there. Can be a long quarantine in a cage, which makes you say they don't want our dog at all. And they have no rabies in those countries. We do have rabies, our dogs get it. The rabies virus needs to get to the brain to kill you. So if it bites you on the toe, the dog bites your toe, injects the rabies virus, it's going to take physical time for that microscopic virus to work its way up the nerve root. It travels in the nerves. And it's going to travel up the nerve all the way through your spinal column and get to the brain. If it bites you on the face, it's going to get there a lot quicker. Now, we do have immunization against rabies, but it is a somewhat dangerous, painful immunization. It doesn't last a long time, so we don't immunize everybody the way we do with tetanus, smallpox, polio. We just immunize those people who are at risk. What we do is we start the immunization as soon as we prove the patient has been bitten and infected by examining, if we can, the autopsy specimen of the animal brain, who also has rabies. And then if you're lucky and get bitten in a hand or a foot, we can probably save you. If you're bitten on the face, it's a whole different problem. We don't have time to raise the antibody response at the first challenge by the vaccine. And so patients bitten on the face or the neck may not survive long enough to have their antibody response. Extremities happen to be where most people are bitten, so most of them do. That's rabies. We need to talk about a few more cells in this system. One of the natural killer cells. We need to bring them in because they're totally different. The natural killer cells are lymphocytes, and they kill a wide range of invaders, but they need no prior exposure. This is really interesting. How do they recognize these foreign invaders. They don't mature in the thymus the way the lymphocytes do. They don't have surface antigen receptors. They live in the spleen, the blood, the bone marrow, the lymph nodes, and they too have to administer the kiss of death. But their recognition is related to something called major histocompatibility molecules. I want to talk to you about those next. I told you how anything you're exposed to in the uterus is not seen as a foreign invader. We have something, and we discovered these when we started transplanting organs, called major histocompatibility antigens. These antigens are on the surface of every single cell in your body except your red blood cells. They don't have any. They are unique glycoproteins, sugar protein combinations, and they are recognized by the body as self. So now you have a situation where you've developed a system of recognizing your own uniqueness. The Australian physiologist Peter Medawar wrote a book called The Uniqueness of the Individual, way back in the middle of the last century, when we were discovering that individuals are in fact unique unless you're an identical twin. There is something on the cell of everybody that says, I'm one of us. And it's literally held up physically on the outside of the cell, so that if you have this killer cell, which doesn't have responses, for example, the way the T cells do for lots of different kinds of antigens, this one has another way of looking. It says, do you have the MHC molecule or not? So the cells hold up that molecule for recognition, and if it's not there, the killer cell will kill it. Same kiss of death, same porphyrins and lymphotoxins, the same mechanism, but what it's looking for is the badge that says I'm self versus the foreign antigens. And that's why it doesn't have to have prior exposure to do this. These natural killer cells are very important. They are present in large numbers where any foreign antigen is likely to invade. So you see them a lot in the respiratory tract, the skin, gastrointestinal tract, urinary tract, anywhere where the foreign invader is likely to get in. And they're very important also in recognizing tissues for transplantation. So what we have had a problem with is, other than in identical twins, getting the body to allow the tissue to be transplanted, because as soon as it's in the body in a very short period of time, this cellular immunity kicks in and attacks the transplant. We call that rejection, and it tries to get rid of it. Now, what happens in those cases is the only way we can get successful transplantation is to suppress the immune system. So we give the system or the patient chemicals which depress the immune system. That makes that patient more susceptible to infection and also to certain tumors which are immune dependent. Not a large number of tumors are, but certain ones are. That brings us to basically through the kinds of immunity in the basic immune system. I would like to show you a little bit about a more fundamental kind you're familiar with called inflammation. We use that term a lot, and the key words here are rubor, tumor, calor, and dolor. Rubor is redness, tumor is mass, like a tumor, calor is heat, and dolor is pain. And those are the 4 signs of inflammation. Here's what happens in this response. If you look at this page, which is showing skin or tissue. In this case, they're actually showing skin because these are the flat cells of the surface of your skin. These are the raincoat on the outer surface, which are waterproofing the skin. And this is the basal layer of the skin, although it's not named that way. Here's tissue injury, a cut, a boo-boo. And inside that cut, microbes, bacteria, have begun to multiply. The body will then, through something called chemotaxis, which are signals that move these cells along, bring in the phagocytes, and also bring in this one, which we mentioned way back in the blood, the polymorphonuclear leukocyte, or a poly as it's called. These are the white cells that get elevated when you have infection. They're elevated in your blood because they come out in masses. And what happens is the blood near that area is signaled to increase its size and flow. It is signaled to increase its permeability. The lining cells of the vessel, called the vascular endothelium, allows the entry of these white cells into the tissue, or the exit from the blood system. Once they go out, they can never come back in. And so you have increased blood flow, the rubor. You have swelling from edema, the dilution. The solution to pollution is dilution. That's the tumor. The calor is the heat from the increased blood. And then we have the dolor, the pain, because you have inflammation and tenderness around that area. That's basic inflammation. Let me tell you a little bit about the kinds of immunity that we see in everyday life. This is another Punnett square showing you about active and passive immunity and specific and non-specific. These are so common that I think it's really worth looking at to kind of wrap this up. We have at the top examples of active and passive immunization. In active immunization, your body is presented with a foreign stranger. The body then mobilizes either the tissue-mediated immunity or the humoral-mediated immunity and attacks the foreign invader. There's an active attack on your part against the invasion. This is done either with cells or antibodies. Both play a part in active immunization, and then you get that anamnestic response if you do it again later. In the passive side, we can instead just give you the antibodies. For hepatitis A, which is usually acquired by eating contaminated food, we can give you something called gamma globulin. That's where the immune antibodies are. We pool this substance from patients, or we used to, and we just give you the antibodies against hepatitis A. That will protect you for maybe 3 to 6 months. You'll eventually destroy them. You don't have any active anti-hepatitis A going on because you haven't been infected, so it'll have to be given to you again. Somewhat dangerous because we have to pool lots of human blood, and we may give you something else, like AIDS or another infection instead. So we don't like to use this too much. Also, another type of passive immunity comes from the mother. The mother has antibodies, and the baby has a very poorly functioning immune system at birth. And so until that baby builds up its immune system, the breastfeeding, which is probably one of the main reasons to breastfeed, is to let those antibodies go across in the milk. This will give the baby protection with those antibodies that the mother has in her blood. That's the difference between active and passive. Now, under both those categories, we also have specific and non-specific. If we look here in active immunization, we can immunize you against smallpox. We actually use cowpox. Varicella, which is related to smallpox, gives us the same response. And in fact, in the 40s, they would bring a cow into the classroom, scratch the side of the cow, take the fluid off, and vaccinate the children. We now do that a little more elegantly. We can use all these different kinds for specific immunity against hepatitis B. We can take killed melanoma cells, make antibodies against them, or let you make the antibodies against them. Oral polio vaccine is another example. So we give you the antigen in some kind of an attenuated form. A killed cancer cell, a cowpox instead of smallpox, which has cross-reaction, or a killed polio virus, or an alive attenuated one, and then you make the antibodies or the cell response that will kill them. We can do a non-specific one. These are two bacteria, Corynebacterium parvum and Bacille calmette-guerin, which is the one against tuberculosis. These do a lousy job at killing those infections, but they are such strong foreign bodies that they elevate the whole immune system. Every cell and mechanism is on high alert, and we give this to patients in an attempt to kill certain tumors, like melanoma, which is a tumor of the skin, And we have some success, not outstanding success, but these are nonspecific. We're elevating your whole immune system. Or we can go to adoptive immunotherapy where we can just give you interleukin-2, which has been incubated with those lymphocytes, the T cells, and we just run that into you, hopefully to kill whatever it is we want to kill. This is usually used, called adoptive immunotherapy against tumor cells. So we have specific, nonspecific, passive, and active forms of immunization. And that's the complete gamut of what we do in the medical world against these diseases. Next time we'll take a look at one of the most widespread and important diseases that we have in society today.